Welcome, students. This is chapter 2 of microbiology, how we see the invisible world. So the main student learning objectives in this chapter is for us to understand the development of the microscope, the history of microscopy, and we'll lightly touch upon the important names and inventors of the microscope. We'll review the different types of microscopes and what it is used for. And we'll also study the staining technique in microscope. A microscope is a lab instrument for seeing very small objects that can't be seen by the naked eye. For example, our eyes can only see about 1 millimeter to 10 millimeters, like a frog egg, but we are unable to see an animal cell or a bacterial cell. So we need a light microscope, which can magnify things at least 1,000-fold. For example, if you want to see a plant and animal cell that's about 100 micrometers, a bacteria which is around 10 micrometers, a light microscope is ideal, a compound light microscope. For anything smaller than a micron, electron microscopy is used when you want to study organelles, a virus, proteins, and so forth. Coming to the history of microscopy, it all started with the discovery of glass by the Romans. So here we have 180. They have discovered the making of glass. That led to the discovery of lenses. And here's Armante, who's used that to make spectacles. And Janssen is also a spectacle maker, but he puts the lens in a tube, thereby laying the way for a current compound light microscopy. Anton Leeuwenhoek is the father of microscopy. He used one lens, so it's called simple microscope in the 1674. And Lister developed on that to make the compound light microscope, and Ruska receives the Nobel Prize for electron microscopy. So before then, they had used light to magnify the specimen, and then they started using electrons. So as throughout history, we can see how resolution has improved, and we are able to seeing smaller and smaller things. Like in an electron microscope, you can see proteins and lipids. Most of the pictures in your textbook is by using the microscope, pictures taken in a microscope. So here is a picture of Anton von Leeuwenhoek, 16th century. He is the first person to observe microbes, like especially bacteria. He used to call them animalcules. And he used just one lens, and there's a lens holder, and this magnified like 300 times, and he was able to describe a lot of things. Then there was Galileo, who discovered the compound microscope, and he used 2 sets of lenses. Robert Hooke developed on it. A little later, again, he wrote a book called Micrographica, and he documented cells of the cock tree. So this is like the word cell is used now. So that's Robert Hooke. So in light microscopy or optical microscope, this involves passing visible light, and the specimen will either reflect it or refract it. So these are through the principles of properties of light and the principles of physics. And then you either use one lens or 2 lenses to magnify the specimen. So in light microscopy that you are familiar with, so we are using light to visualize images. And there are many subcategories here. There is bright field, dark field, phase contrast, differential interference, DIC, fluorescent microscopy. There's confocal scanning laser microscopy, photon microscopy. So there are many light microscopic techniques and tools that are currently used. So we have different types of microscopy to complement each other in diagnosis and for doing research. So here's the light microscope, and then here's the oculus through which you would look at. It's a compound light microscope because it uses two sets of lenses. There's lens here in the ocular, and then there's a selection of lenses near the object. So they're called objective lenses in a revolving nose piece. The arm of the microscope, the body tube, the mechanical stage clips where you'll keep the slide and then anchor the slide in these clips. We have the coarse focus knob, fine focus knob, and here we have the light source in the base of the microscope. The light will pass through a condenser, and the diaphragm can be used to allow adjust the amount of light. The light will fall on the specimen that's kept here, and the image will be magnified through these set of lenses. Some of the terminology that we had studied was the field of view. That is the circle of light that you would see on this of the specimen, field of view, depth of field, total magnification, which is the magnification of the ocular lens and the objective lens. So you can either use the scanning, the intermediate, the high power, and there's also an oil immersion in our labs. Most of the microbes are colorless, so we will add a dye, which is called as a stain, in order to increase the contrast and resolution. The solving power or resolution of a microscope refers to the clarity. So two close by objects, right, are seen as two separate objects when there's high resolution. Usually, if we use a shorter wavelength energy to light up the sample, the resolution is much high, like electron microscopy. In bright field microscopy, the specimen is usually fixed, and then it is stained, and then it will appear dark in a light background. And this is very widely used to observe bacteria, the morphology and the structure in bacteriology. In oil immersion microscopy, so this is a technique that we use to increase the resolving power of the microscope. So what we'll do is we'll replace the gap, the air gap between the objective lens and the cover slip with a highly refractive oil. So what that does is it prevents scattering of light as it passes from below the slide to above the slide. So this in turn can produce a very sharp and clear and high resolution image. So that is oil immersion microscopy. The next microscopy technique is dark field microscopy. So here the sample is not stained. And here the sample will look bright against a dark background. So this is achieved by placing an opaque disc between the light source and the condenser that will block most of the light. And the light is scattered off the specimen and it is reflected to produce these images. So the advantages is there's no stain required, and you can use a live specimen here. And it's widely used to study spirochetes in microscope, in microbiology. It's like a screw-like microbe. So treponema is the spirochet that causes syphilis, and Borrelia causes Lyme's disease. Phase contrast microscope would be next. So here in phase contrast microscope, so this is a technique that converts a phase shift in light passing through a transparent specimen to brightness changes of the image. So the phase shift is invisible, but it will become visible when shown as a brightness variation. So this creating contrast. So this is based on the principle that a small phase change in the light rays, it's induced by differences in the thickness and refractive index of the different parts of the object, can be transformed into differences in brightness or light intensity. So this is very widely used to study eukaryotic cells when we want to study the internal structure of a cell. So this gives a higher resolution and contrast without staining. Differential interference contrast microscopy, or DIC. So this technique will produce contrast by visually displaying the refractive index gradient of different areas of the specimen. So this process will begin with light from an incandescent light source passing through a filter that is placed between the light source and the condenser. Then it uses interference patterns of the beams of light that differ and produce a high contrast 3D image. And this is widely used for studying structures inside a live cell without staining. So here's a picture of a fungus that causes a skin disorder. It's called Fonsecaceae. In fluorescent microscopy, fluorescent dyes will be used. They are called fluorescent chromophore or fluorochromes that are capable of absorbing energy from the light source and emitting the energy as visible light. So we are going to use a fluorescent dye and also special like UV lamp to light up the specimen. And this is very useful for studying proteins inside a cell. So here's some kind of an immunology that's going on with antigens and antibodies that can be visualized with fluorescent microscopy. It's very sensitive, specific, reliable, and this is made for observing the localization of molecules inside a cell. Confocal microscopy uses light from a laser through the objective of a standard light microscope to excite the specimen within a narrow plane of focus. So any emission of light out of the plane will be rejected by the pinhole, and it will scan a lot of planes, and the two-dimensional images will be put together to form a three-dimensional image. And this is ideal for studying thick specimens like biofilms. Here's an electron microscope. So here, instead of using light, it uses electron beams. Electron beams have a much shorter wavelength compared to visible light. And hence, there is very high resolution and magnification, ideal for studying subcellular structures. There's two main types. In scanning electron microscope, you can scan the outside of the organism and study the topography and texture of the organism. In transmission, so these are thin slices of the specimen, so you can see all the internal structures. So in transmission electron microscopy, the electron beams are aimed at a very thin section of the specimen, and the specimen is usually like stained with like gold particles to enhance contrast. And then the image is focused into a viewing screen of film, and it can amplify up to 500,000 times. In scanning electron microscopy, you study the outside surface of the specimen. The electron beams scan the surface of the specimen that is coated with gold. It has excellent depth of field, and this can amplify about 30,000 times. The most sophisticated microscopy would be the scanning tunneling microscope, where there'll be a probe that passes above the specimen. A voltage bias will cause an electric current, and the intensity is based on the distance between the probe and the specimen. So these are very, very high resolution for studying atomic structures. Atomic force microscopy, here the probe is also passed above the specimen. This measures the variations of the current, and the resolution is atomic level. So the next two slides are summary slides of the different type of microscopy. So it's talking about the different types of microscope, what is it used for, and a sample of the images. What is the difference between transmission electron microscope and scanning and atomic force microscope? So these are summary slides. So how do you prepare a specimen for staining? So cell staining is a technique that is used for visualizing the cell in a microscope. A wet mount is a technique where you will take a drop of a fluid containing the organism in a slide. And then a smear is, you would take a loop full of microorganisms, and then you would spread it on a glass slide. And then you fix the microbe. It's called heat fixation, so that it can adhere to the slide and then it can accept the stain. So we have different types of staining for different type of microbes, depending mostly on the cell wall structure. See, there's thousands and thousands of microscopes. In order to identify them for diagnosis and treatment and for studying, these are valuable tools in microscopy. So some of the dyes that we'll come across are basic dye, which has a positive charge. A dye is also called as a chromophore. Chromo means colored. Acid dye has a negative charge. In positive staining, the stain will be absorbed by the cell and you can see it. So that is called positive. Whereas in negative, the cells won't absorb it, but you will see the stain in the background. When you say simple stain, we are using only one dye that will reveal the basic structure and the shape and arrangement of the microbe. In differential, we'll use two or more dyes that will distinguish between the organisms, like gram stain, acid-fast stains. So here is the protocol how a differential stain works. So in gram staining, so you have a mixture of both gram-positive and gram-negative bacteria that differ in their cell wall structure. So the first step is when you take a fixed smear and you add crystal violet to the smear, both the cells are purple in color. And then the next step is to add iodine, which is a mordant that will make the dye less soluble and it adheres to the cell walls. Again, both the cells are purple. And then when you decolorize it with alcohol, gram-positive will retain the stain and gram-negative will lose the stain. And now you have to do a counter stain in step 4 with safranin, which will stain the gram-negative into this pink-red color. Some terminology is the primary stain that will give all the cells the color. A mordant will stabilize the stain, and iodine was used here. The decolorizing agent will remove the color, remove the extra color from the peptidoglycan wall. usually alcohol is used for that. A counter stain here was safranin, which will stain the decolorized cells from the step before. So there's gram positive that is going to retain the crystal violet stain, gram negative that doesn't, it will light up with the safranin. There are also gram non-reactive that does not stain or stain poorly, and gram variable, which will stain very unevenly. Some other stains are the Zeal-Neeson acid-fast stain. This is widely used for detecting mycobacterium that causes leprosy and also TB, Mycobacterium tuberculosis. So this is a protocol because they have mycolic acid protective layer. So there's a special acid-fast stain to visualize that. There's a negative stain for visualizing capsules. The background around the organism is filled with a stain like India ink or negrosin or acidic dye. In flagella staining, a mordant is used to coat the flagella and make it visible to see, and then iodine will be added repeatedly. For visualizing endospores, the heat fixed smears are covered with malachite green, and then they are heated until they are steamed for 5 minutes to make the wall more permeable. Then they will add the malachite green, then it's rinsed off, and then the counterstain safranin is added. So we have a range of staining techniques and to visualize microbes for proper diagnosis and treatment. So these last two slides are summary slides of the different stains that we'll study in lab, the dyes and the techniques. And I hope that was helpful.